My name is Oliver Fien. I am Director of the West Coast Metabolomics Center. I welcome all attendees to our this month's webinar. We have a distinguished guest here, Dr. Evelyn Rampler. She is a group leader at the Department of Analytical Chemistry at the Faculty of Chemistry at the University of Vienna in beautiful Austria. Dr. Rampler did her PhD in 2013 as a corporate PhD thesis in the Division of Analytical Chemistry and Bioman Research Center in Austria. And before, she had a bachelor in food science and biotechnology and a master in biotechnology at the University of Natural Resources and Life Sciences in Vienna. She did a habilitation, which is a European thing, to kind of like become a professor in 2021, multi-omics analysis using LCMS in analytical chemistry. Before she had a permanent senior scientist position and research stay at Thermo Fisher in Bremen, as well as at the ISIS in Dortmund, a lipidomics research group of Robert Ahrens, and an environmental analysis group at University of Vienna, a postdoc fellow in Biocenter Vienna, and also a glycomics research group at University of Georgia in Athens in USA in 2013. So she is leading a group now with a focus on glycolipids. Glycolipids become ever more important. There's always more papers coming out showing the importance of glycolipids as well as modified glycolipids in all sorts of diseases, including Alzheimer's. And that's why we want to learn more about it. Because we also sometimes find like glycolipids that we think like, this is not possible to have these things, and what do they do there? And are we sure that we identified them right? And therefore we want to learn from Evelyn today how to do it right. Evelyn, the floor is yours. Thank you for joining.
Thanks for the kind introduction, Oliver. So yeah, thanks again for the kind invitation. And I would also like to thank Janet Martins for the organization of the talk. It's a great pleasure for me that I can speak here on the online WCMC guest lecture seminar series at the UC Davis. And I will talk about new avenues in high resolution mass spectrometry-based lipidomics and glycolipidomics. So not only glycolipids, but also a bit about our lipid pathways, workflows. And before I jump in the actual research part, I want to shortly give you some background about my lab. So we are located in Austria. The capital is Vienna, and it's the heart of Europe. The University of Vienna is a very old and very big university. We are 657 years old and have around 1,000 people, including students and staff. The Faculty of Chemistry is in the center of Vienna, and my lab is located there. We have a strong focus on using high-resolution mass spectrometry instruments in combination with liquid chromatography. So most of the data I will show you today is from the IDX from Thermo, having an orbit trap and an ion trap. So we can do multi-stage fragmentation. Please keep that in mind. I will return to that later. And we combine it here with the UPLC Wagfish system. Our newest requirement is ABSAC Xenotoph, but this was just installed last month. And my lab was founded only a year ago. We are a rather small but constantly growing group with the research topics of developing novel SMS-based omics assays, a strong focus on glycolipidomics, and we have also a big FWF funded, that's the Austrian Science Funds funded project on lipid-based communication with the tumor microenvironment. So I'm interested in lipids, lipidomics, and we are looking, when we are looking into the omics realms, which are going from DNA over RNA, proteins, metabolites, and all kinds of subsets such as lipids and glycolipids. It's clear that we need powerful analytical techniques to look into these molecular subsets. For example, for genomics and transcriptomics, it's next generation sequencing. But for the post-genomics approaches, we all know high resolution mass spectrometry is frequently used. And when we want to analyze lipids, looking into the human metabolome database, there are 250,000 metabolites annotated so far. we can already see the importance of lipids because 80% of these annotated metabolites are lipids, and there are still some glycolipids and other classes for sure missing. The analytical challenges here are the structural diversity. We have different physical chemical properties. Many of them, for example, are amphiphilic, so we need tailored extraction techniques, stability issues with oxidation, high dynamic range, with low concentration, high concentration lipids in the same sample, and of course, isomeric and isobaric species. And when we want to deal with this challenge, high resolution mass spectrometry in combination with liquid chromatography is ideal to deal with these challenges. What we have then, we can combine the mass analyzer, the power of the mass analyzer with the chromatographic system, meaning the retention time information we get here with the mass to charge information and fragment information. Depending on the energy we are applying, we can either have a lot of fragments or just some of them. And all this LCMS-based omics workflow, independent now, if you're analyzing peptides, lipids, or metabolites, include one sample collection step, an extraction of the analytes, the analysis by LCMS, which can be targeted or non-targeted. So meaning that we can have a specific subset, such as glycolipids classes we are looking at, or we want to screen everything inside the sample. Then, as you all know, probably a tedious data interpretation step. And in the end, we can have either this identification list, so IDs as a result, or a quantitative result. For MS-based lipid and metabolite identification, what we need to consider is the quality of annotation, which can be that we have a low level of information with only the mass to charge ratio. The Metabolomic Society recommended to use these categories A to G. So a category G is the lowest one with the unknown formula. And we can go all the way up to known enantiomer information. For high resolution mass spectrometry, we have this, we are usually in this area that we know a functional group, or we could, for example, see the molecular species level of the lipid, which means we can read out the different fatty acid chains which are bound on this phosphatidylcholine. If you want to know more, I can recommend reading our recent review on recurrent topics in mass spectrometry-based metabolomics and lipidomics in analytical chemistry. Now for quantification, we developed a special workflow to deal with the challenge of quantification in high resolution mass spectrometry based on labeled yeast. This was done in cooperation with the Kölnsberger lab and Isotopic Solution. What we do here is we have a uniformly labeled glucose, which is then fed into the fermentation system. And you have this Pichia pastoris yeast strain, which is fed by this glucose. And after a while, everything inside the cell will be completely C13 labeled. So when we harvest and do chemical extraction, the proteins, metabolites, and lipids, they're all C13 labeled. Meaning we can use them for quantification because they have this shift in mass to charge. And that means you can account for all the error accumulated during the analytical chain of sample preparation, storage, and measurement. I was specifically involved in doing the lipidome isotope labeling of yeast, the Lilly extract, so the lipid extract. We did all kind of characterization and quantification studies in the way of commercializing this. And this we're then using on the one hand, spiking into a sample for quantification, but we are also using it to benchmark our mass spectrometry instruments. So comparing the daily performances, and that can be done for both metabolomics and lipidomics, depending on the extract we are using. So for the ethanolic yeast extract, we have identified so far 210 C13 labeled metabolites, which we can use for quantitation and benchmarking. And for the Lilly extract, that's a chloroformic yeast extract, we are at 420 C13 labeled lipids, which we identified so far. And these two extracts are also are or will be available with our partners of Cambridge Isotopes and Cayman. And yeah, I really think it's a powerful tool for quantitative issues when dealing with metabolomics and lipidomics. For glycolipids, I have to say they're not, for example, no gangliosides in yeast extract, so it's limited for the glycolipid classes, but for lipid bulk analysis and standard metabolites, it's very powerful. So now we talked about how we are doing identification and quantification. I want to go into the methodological developments we did to increase coverage and throughput for the lipid analysis. This is an example where we combined hydrophilic interaction and reverse phase chromatography with high resolution mass spectrometry. What we do here is inject the sample on the hydrophilic interaction column. And then all the polar lipids are sticking to the Hilli column. All the non-polar lipids will walk through and will be trapped on the serial coupled reverse phase column. Then we have this 6-port valve, which enables us to have another pump with a different Evelyn system and then elute the non-polar lipids from the reverse phase column all together then in the mass spectrometer. So using this technique, we can measure polar and non-polar lipids in 10 minutes. With a calibration time, we are here at 15 minutes overall method for the method timing. We were able to identify, for example, 400 lipids in human plasma SM9050 with this method. And what is nice here, when you do new identification, you have an additional polarity assignment. The second method is something which is basically the state-of-the-art method in our lab, where we're using reverse phase chromatography in combination with high-resolution and multi-stage high-resolution mass spectrometry and multi-stage fragmentation. In the lipidomics field, most people doing reverse phase are having this isopropanol gradient and mostly using C18 columns. We also use C18 columns, but this example here, I show you a C30 material, so C30 column, which makes it possible that you have a better lipid isomeric separation. What you can do here is measure the major lipid classes in 28 minutes. That means that we can get all the bulk lipids here and additional structural information by the MS to the M, the MS3, for example, for triglycerides assignment. So we use here the parallel ion trap and orbit trap fragmentation for lipids in complex samples. And I want to emphasize, when we're using this workflow, we are also spiking in the little lipids so that we could merge the profiling and quantification in one analytical workflow. That's quite powerful if you do standard bulk lipid analysis. I now want to jump into the applications and ongoing projects. I want to show you one example for the lipid profiling and quantitation of bulk lipids in colon cancer cells. And then I chose 2 examples from the glycolipidomics realms, where we have the gangliosides as differentiation markers for human mesenchymal stem cells, and the so-called glycosyl inositol phosphoceramides, which are plant glycolipids, and we want to use them to monitor heat stress in different plants. So for the first bulk lipid analysis example, this is in the area of the lipid-based communication with the tumor microenvironment project. Here I'm the lipidomics package leader, and Katharina Hohenwallner is my PhD student in this area. So why lipids in cancer? You're probably aware that the lipid metabolism is crucial in the pathogenesis of cancer in humans. And we want to use the altered lipidome to investigate the cancer aggressiveness and response to therapy of anti-cancer metal drugs. It was, for example, shown that there's an accumulation of lipid droplets in colon cancer, and this is a central component of therapy failure with standard chemotherapy, including oxaliplatin. So the example I chose here, we are measuring colon cancer cells which were treated with lipid regulators. We have these colon cancer cells treated with this ruthenium drug, and we have then a sensitive cell line and a resistant cell line. The resistant cell line has the increased lipid droplets, which you can see here. This nice green spots are increased, that are the lipid droplets. And we wanted to look into the changed lipidome here. further treated the cells with oleic acid, which is known to increase the lipid droplet formation. Trioxin C should inhibit the lipid droplet formation, and the DMSO is our control. So what we saw with this reverse phase MS to the N workflow I showed you before is the possibility to measure 250 different lipids of 16 classes. We used here also the AcquireX tool from which is available on the Thermo machine, where we can use automatic inclusion and exclusion lists to do deep lipid profiling. And by spiking in the Lilly lipids, we could quantify 88 different lipids from 7 classes. And what you can see now, looking at the different classes, is there are some classes which are basically not changing. For example, phosphatidylicholine. So the standard plasma membrane lipids seem to be not changing too much. However, there are huge changes for the triglyceride and also for the diglyceride level, as you can see here, specifically when you're treating them with the oleic acid. So I think it shows the powerful, it's a powerful technique to doing the standard bulk lipid analysis. And we could further see here group separation of the treatment from oleic acid with trioxin C and DMSO. Unfortunately, the downregulation of the lipid droplets with trioxin C was not significantly changed compared to the DMSO control, but we saw this regulation with oleic acid, and we could also have some triglyceride markers, which we are currently looking further into how to use them. So this was an example of showing this lipid quantification, identification of bulk lipids with the Lilly extract, and I now want to jump into the actual glycolipid analysis. I'm not sure if you know the song Bittersweet Symphony from the Verve. This is what I'm thinking of when I'm analyzing my glycolipids. This is why I'm calling them Fatty Sweet Symphony of cells, where we are looking into these nice glycolipids. They are, chemically speaking, they have this fat hydrophobic part, you can see here, and the sugar polar part, which is usually pointing towards the outside of the cell. So you can already see they're amphiphilic, they're rather difficult to, or you have to think about that in the extraction process. And then there's the problem of lacking standards, which we are having, and also annotation workflows. So as you probably all know, Karen Bertozzi recently received great recognition with the Nobel Prize in Chemistry for bioorthogonal labeling of glycans. That makes me, on the one hand, very happy because she's the 8th woman in chemistry, only the 8th woman in chemistry who got the chemistry prize in Nobel history. And she's also a shining model for diversity in research, which I think it's very important. But it also highlights, most importantly here, the need for pioneering method development in glycan sciences. So traditionally, what was done for the glycolipid analysis was chopping off this glycan part, for example, for these glycolipids found in the cell, and then you would only analyze the sugars there. But we want to really do the whole glycolipid, so I definitely think it's glycolipid time. The methods are ready to do that, but we need to bridge the gaps between mass spectrometry-based glycomics and lipidomics. So the people were analyzing it from the glycan part or looking at the lipids only. And for annotation, this is of course important because we need some way of reading it out when we have the signals, when we extracted that. And I want to show you now some work, 2 specific classes and how we were doing the method development for the gangliosides and the GRPC. Yeah, gangliosides are sphingolipids, acidic glycosphingolipids. Again, they have this glycan moiety. That's the one which is outside of the cell, and the ceramide moiety, which is found inside the plasma membrane. What is classical for the ganglioside that they have at least one sialic acid. And for the amphiphilic character, we need to consider that for the extraction. The problem is where not a lot of standards available for gangliosides. At least there are some, but not many, especially when it comes to quantitative standards so that we know the concentrations. And there is a lack of databases. Some databases can give you values, but usually they don't cover all the ganglioside classes. So this is why we also went to do our own annotation workflow here. For us, the gangliosides were interesting in the project, going from mesenchymal stem cells to differentiated cell types, where we wanted to look into the gangliosides in these different cells. The workflow we were applying here was built by my students Nina Troppmeier and Katrina Hohenwallner, and I will now walk you through the sample preparation, data acquisition, and data evaluation steps. So for the sample preparation, we had these mesenchymal stem cells, where we're coming from adipose tissue, from surgical procedures, and our partners from the University of Vienna Natural Resources, University of Natural Resources and Life Sciences, they were extracting the mesenchymal stem cells, expanding, isolating them, and then differentiating them into chondrocytes, adipocytes, and osteocytes. And we then got these samples that spiked in deuterated gangliosides, which were available in known concentrations, to do at least some kind of quantitative assessment, and then use the MTB extraction, so this two-phase extraction. And in this case, we have the glycolipids also in the lipid phase. Yes, we see all the gangliosides here in the lipid phase. We couldn't see anything in the metabolite phase. And with this extract dried down, and then in the starting conditions from the ALC column, we started to do the liquid chromatography and mass spectrometry workflow, where we have the Vanquish UPLC system and the Orbitrap IDX. So again, we have here the parallelizable mass analyzer, which we can use. So that means we have the Orbitrap, we can do high collisional dissociation, and the ion trap, which can do collisional induced dissociation. And for the IDX, what we do, we have a mass list where we generated all classes we were interested in, and the different possibilities of hydroxylation states and lipid lengths. We ran this as a trigger list, and whenever we see something of this list, an MS2 trigger will be started in the Orbitrap. And when this is confirmed to be a ganglioside, for example, you see a sialic acid loss of 290, in parallel, we run this MS3 fragmentations to get more structural information about the ganglioside. So we get a lot of specific fragments for the ganglioside, a lot of structural information. And here I want to emphasize the difference between lipid class and molecular lipid species level annotation. So many state-of-the-art methods, especially the ones with antibodies, for example, will only read out the lipid class. So they know this sugar part, it's a GM3. But when we have the lipid species level, we know also the length of the lipid here and the hydroxylation state. And the thing we can do now is reading out the molecular lipid species. So meaning also having the long chain base information, fatty acid information. For the annotation, what we use is the lipid data analyzer. It's a tool which is really available and which is based on decision rule sets. So we use the information about the fragments to train our annotation workflow, and then we can go for the samples and annotate them. This we did in cooperation with the University of Graz, with Jürgen Hartler's group. And yeah, what we have the retention time information from the reverse phase column. We have the MS1 information, MS2 and MS3 information, the fragments, and then we use this to identify our gangliosides based on the fragmentation pattern. We also use the specific filtering steps here with self-written R scripts, considering the equivalent carbon number model. So longer lipids or more hydrophobic lipids will stick longer on the reverse phase column, and these rules need to be followed. Otherwise, we kick out the hits we are getting. We are setting intensity threshold and also MS2, MS3 spectral quality. Here I want to show you one example why it's beneficial to have the combination of chromatography and multi-stage fragmentation, and why I won't do shotgun analysis with glycolipids, because we have a huge structural diversity. For example, this GM3 you can see here has a difference in the double bond positions, and it can be separated solely based on the reversed phase chromatographic separation. So you see these two peaks here. And when we do now the MS part, normally in negative mode, you see the glycosidic fragments of the glycolipid, and in the positive ion mode, mostly ceramide fragments. But by combining now this MS3 information or adding this up, we get more structural identification or more structural information of the glycolipid. And so this helps us to do a correct molecular lipid species level assignment. So what we got now in our samples from the human mesenchymal stem cells and differentiation product is we could separate them by their according to the cell state, or not cell state, but cell type. And this is solely based on the ganglioside profile. When we did the bulk lipid analysis, this was not possible. So it's really something in the gangliosides differing on the surfaces of the cells. That's very interesting. This is when we are using the species and molecular lipid species levels. When we are looking in the lipid class level, where most of the state-of-the-art methods have the readout here, you can see that it's not really possible to have one specific marker. You can maybe see certain trends. For the osteocytes, we have higher sugar series compared to the other cell lines, but basically the differentiation based on the ganglioside profile is only possible when we consider species and molecular species information. So in our samples, we found 137 unique gangliosides in the MSCs and differentiated cells. 78 of them were potential differentiation markers, which we could identify. And what was really interesting, we had really 48 completely new gangliosides. Completely new means based on the molecular species level. And we could have a quantitative assessment because we had the deuterated standards inside there. So we can at least say we are here in the nanomole to micromolar range and can compare the different groups because that helps you for the relative quantification of the gangliosides. The highest amount of gangliosides was the GM3 group, followed by GD3 and GM2. I want to show you now one specific example of markers we were identifying. What was interesting here, it's really an isomeric marker. So meaning depending on the molecular lipid species level, it's different in the different cell types. So this GM3 having a long chain base with 18-1 and the dihydro is dihydroxylated and fatty acids of 24-1. Is upregulated in the adipocytes compared to all the other cell types, whereas the other one with 2 double bonds on the long chain base is upregulated in the osteocytes, meaning that they are also chromatographic separated here, which you can nicely see. We really need to consider these different structural levels of gangliosides for our analysis workflows. It matters. So for this new glycolipidomics profiling strategy, we could develop for ganglioside and glycolipid analysis. We were able to use reverse phase high resolution MSN for the annotation, get in-depth structure characterization up to the molecular lipid species level. Of course, I have to say here, it's not possible for all the lipids to see the molecular lipid species, because if they're low concentrated, you will not be able to see the MS3. But in this case, we always have at least the lipid species level. So it's better than just the lipid class. So we have a lot of structural information here. We do believe, because we can see this ganglioside pattern helping us to differentiate the cell types, that they might be possible to use as additional class of differentiation markers, so-called CD markers, we might be really able to see here differentiated ligand presentation of this constitutive signaling of different cell types. I think that's biologically very relevant, and we need to look into that in more detail. And currently we are further optimizing and implementing it to other glycolipids. This paper just got published last week in Czech. This is why I put Czech's Gold, sorry. Put the QR code here. So if you want to have more information, you can just go and visit the page there. We also have the data freely available and a link to the lipid data analyzer download, so you can use our workflow and work with that. The second glycolipid class I want to show you we are looking into is glycosyl-inositol phosphoceramides, so-called GIPCs. Most people haven't heard of them, but they're actually the major sphingolipid on Earth because they are present in plants and algae in the plasma membrane. So they're very, very abundant, but it's very difficult to analyze them because at the moment there are no standards at all. But they're important in terms of biology because they're they're involved in plasma membrane growth, cell signaling, organization of membrane, and host pathogen interaction. For us, most importantly, they are also involved in response to stress and heat stress. In terms of chemistry, looking into the chemical structure of the GIPCs, on the first side, they look quite similar to the gangliosides, only that they have this inositol phosphate and no sialic acid present. But in terms of amphiphilic chemistry, they're quite similar. We had here this project together with the Federal Institute of Enology and Pulmonology in Austria, the Medical University and University of Vienna, where we tried to have our model systems with Bali and Arabidopsis to develop our plant-based GIPC extraction strategies, and that we then identified drought and heat specific GIPCs markers, and we want to transfer that then to analyze Grüne Veltliner. This is a white wine, very common in Austria, a cultural acid basically, and due to the rising temperatures of climate change, it's in danger. So it would be very important to have heat stress markers here. And what we did now to develop our GIPC method is we used this approach very similar to the one I showed you for the gangliosides. Only we needed to have a different extraction strategy here. We have here isopropanol hexane water mixture. And because we are dealing with plants, starch is a problem. We are also doing alkaline hydrolysis to get rid of the starch here. And then we have a reverse with high resolution MS to the N workflow comparable to the one with the gangliosides, again using the parallelizable mass analyzer capacity to do multi-stage fragmentation. We then had this decision rules again and did that for the lipid data analyzer to assign our GIPCs. This work was done by my students Lisa Panzenberg and Nina Thür. And now when you want to annotate GIPCs, it's very problematic because here are no commercially available standards at all. So how to do a proper annotation about something without a standards, that's quite tricky. So what we do is basically get every information we can use and then try to implement it and check our methods. So we use, of course, the high mass accuracy of the mass analyzer, then fragmentation patterns. We use matching retention times in positive and negative ionization mode. So we only set true hits if we find it both in positive and negative mode. We use the equivalent carbon number model because we have, again, the reverse phase column here. We know more hydrophobic things have to elute later using these retention time trends to kick out false positives. And then, of course, all the literature information we can get. Here you can see a simplified model of the GIPCs. They can have only little sugar attached for the A series, and it can get quite complex up to the series E, as you can see here. Here I want to show you, because it's such a beautiful, complicated class, some fragmentation pattern. Here you can see a GIPC, which is from the A series with the amine residue in the negative ionization mode. You can find in the negative mode the specific fragments of the inositol phosphate and its water loss, so 259 and 241. And this can be then used to assign our GAPCs. As an example for positive mode, I show you a more complex sugar series from the D series and trihydroxylated. Here you can see that the B series ions are quite abundant in this case. And yeah, now using the fragmentation pattern, we went into our samples. So we started with the spring barley as a model system, and also because it was of interest for our cooperation partner from Verena Eber's lab. And we had here spring barley grown for 6 to 8 days and 10 to 12 days. We applied heat stress for one part of the samples with 42 degrees, and the other one was run at standard 20-degree control. And then we looked into the GAPCs and what was going on in the barley grains. So looking now into the GAPC change upon heat stress, you can see that the red one is always heat stress, green is control, that the control versus heat stress, really heat stress has increased GAPCs. But interestingly, it was more pronounced in the early development stage compared to the late development stage. We could, by this data, confirm what we saw in the literature, or what they predicted in the literature, that GAPC should be accumulated upon heat and drought stress. They are also saying that's probably due to the fact that they're trying to increase the sugar contents on the plasma membrane to increase the desiccation tolerance. It probably makes sense. And using now this workflow for the GAPCs we developed in the barley, we went into this wine samples, the Grüner Veltliner samples. Here we are on the sampling site in Klosterneuburg. At the moment, we could already detect several GAPCs in wine leaves, so the method is working. And in summer, what we did, we had a heat stress experiment where we had some of the wine plants were having a foil on top of it, so there was much more heat applied. And then we had the normal wine growing and compared this as a heat stress experiment. But at the moment, we already collected the samples, but we are in the process of analyzing them. So in a nutshell, I'm already coming to my conclusions that I hope I could convince you that the combination of liquid chromatography and mass spectrometry with high resolution mass spectrometry with multi-stage fragmentation is powerful for doing both identification and also quantification of lipids and glycolipids. We have we can do complex structural analysis by multi-stage fragmentation. We can use the labeled little lipids to merge lipid identification and quantification. But as I said, this is more for the bulk lipids. For the glycolipids, there are still limitations with the classes available. But what we could do, we developed the first automated annotation tool for several glycolipid classes based on lipid data analyzer, which you can now use. It's ready to go, and we are currently extending it. We did analyze lipids and glycolipids in complex samples. So I showed you the example of the colon cancer cells, where we had the bulk lipids we've analyzed, and then for the glycolipids, we have the gangliocytes in stem cells, where we showed that the differentiation, that there are markers for the differentiation states, and also for the GAPCs, that they are potential heat stress markers. So in my opinion, we are now able to deep dive into the fatty sweet symphony of cells. And in future, what we are aiming to do is doing complex structural glycolipidomics, more on the glycolipidomic side, a bit on the lipid side, of course. And we are using the combination of high resolution and liquid chromatography. And we are also assessing whether UVPD and EAD fragmentation will help for the electron activated dissociation. We are very much interested to test what the CENOTOF can do for us. And with that, I'm already coming to my acknowledgements. I want to thank all my colleagues from industry and academia, national and international cooperation partners. I want to especially thank all the members of the Kölnsberger and my lab. It's basically we are basically acting as one big group, and it's a very fruitful cooperation environment. I also want to thank all the students of which I presented the work here and the funding agencies. I also want to emphasize the female mass spectrometry here. It's a very supportive international group. And I want to thank always my family for supporting me, that I can still do a bit while I'm on maternity leave. Here you can see my group visiting me at home. That's my daughter Annalie in the garden. And you can see we have a lot of fun, and it's really great to work with them and that they can basically run the lab while I'm gone. I'm very lucky to have such a great team. And with that, I'm thanking you for your attention, and I will be very happy to get any questions and discuss with you what's the future of lipidomics and glycolipidomics.
Thank you, Evelyn. It was fantastic. As usual, Dr. Arpana Vania will guide us through those questions, and then we'll see how we continue. Okay?
Great. Thanks, Oliver. Such a great talk, Evelyn. And I also wanted to say congratulations for making the top 40 under 40 analytical chemistry list. If you haven't seen that, that's really great. That's such a great achievement. So with that, I'll start with questions. Among the hexosyl ceramides in your Hillock method, would the glucosyl ceramide and galactose ceramides also separate?
No, the glucosyl and galactosyl ceramides will not separate, but if I have dimeric and monomeric ceramides, then they will separate. That's the problem always when you're doing mass spectrometry, that it doesn't matter if it's galactose or glucose. Yeah, that's quite tricky still to do. Maybe we can do more with EAD or UVPD in future, let's see. But then it's only about where the fragmentation, where it's fragmenting and having standards available. But yeah, super tricky.
Yeah, that can be, it's still a very big challenge, in terms of separation. Is the data analysis for the C13 labeling profile and quantitation done by an in-house tool or any endorsed open access tool?
Good question. So when you mean the for the quantitation, or because for quantitation we're basically using Skyline, so we have a target list in Skyline, and this list we have this benchmarking paper where you can export the list and use it, so and import it in Skyline and you're ready to go. But in terms of the quantification, when we are using it for, we also have a way of doing flow injection, a combination with reversed phase. This is not published right now, and we still do with an R script. Yeah, but we describe it how to do it in the paper.
Okay.
Yeah.
And after I assume after the paper is out, you'll make that a script available.
Yeah.
Okay.
That's
Wonderful.
always what we want to do. But whenever you have a targeted list and you know what you're analyzing, then it's not a problem at all. It's more for the non-targeted, targeted, where you, even then you could get the target list and then apply it. So yeah, it depends on what you want to do. Most of it is available, but there are certain things you still, we still cannot just download.
Great. You had showed that you use LDA for MS to the N data annotation. Is there a reason why you don't also use CD or Compound Discover for data annotation or data processing?
Why I didn't use Compound Discover for the processing of the
MS to the N, MS to the N data.
So you mean this module, so as far as I know, what they could do is just do fragmentation trees, but what I want to do is a non-targeted workflow, or not non-targeted, targeted. So I want to see the gangliosides analyzed in all in my sample. So I need something which considers the specific fragments, and this cannot be done with compound discoverer. So that's why we go for lipid data analyzer. I think Oliver also had some earlier from 10 years ago ganglioside path workflows which could be used. But for MSN data, as far as I know, this is the only way of doing it right now.
Okay. For ganglioside isomers, is MS⁻³ enough to differentiate the lipids? Have you tested anything beyond N equals 3, or do you go beyond MS⁻³? Is that even like necessary? I always ask this question because I'm interested in MS⁻N from the flavonoids side of things. So I just kind of want to hear your opinion on that.
No, it's a good question. So problem with, or the benefit of MSN, of course, is to get more fragment information, but it's always limited to the concentration of your glycolipids and gangliosides you're analyzing. So for me, going farther than MS⁻³ is mostly not possible because it's not enough concentrated. But if you would have, I don't know, a sample which is highly concentrated, you could definitely go and add MS⁻². But this is here somehow a middle way of doing targeted, non-targeted ganglioside analysis in samples, and it made sense for us to add the MS3, but MS4 there would have not enough signals to add that, I would say.
Yeah, that's definitely a huge problem.
Especially with the LC combinations. I always want to have the LC part. I rather have the retention time addition when MS⁻N and shotgun.
Right. Yeah. It's so dependent on the number of ions that you're seeing in MS⁻⁻⁻, and that really kind of determines everything else down the road.
That was the right the number of ions was the right term for explaining. There was another part you were asking, but I forgot it.
Have you tested? Well, I guess I think you've answered it. Like if you tested beyond MS⁻³ really.
It's just too low intensity. So the ions we have is, it didn't make sense for us.
Okay. Have you done it with maybe your standards, or like when you're acquiring library data?
With the standards, the problem with gangliosides at the moment, there are only 2 isopore standards, deuterated one available, which we spiked in, and the rest is always mixes. So meaning not good. So I have, for example, a GM3 ganglioside standard, but that means I have all kind of different lipids parts on it. So it's pure in terms of the lipid class, in terms of the lipid class, but not in terms of the molecular lipid species and species level.
Okay, and I'll go to this question that's kind of related to uploading data. Have you uploaded any of your MS or MS⁻³ spectra of your ganglioside lipids to some repository like massbank.us? I mean, you just said that there's 2 standards, but have you uploaded them?
Yeah, so with the paper which was just published in JACSCO last week, we add all our raw data to GMPS, and there's a link. So if you're interested in that, you can find all the data there. Oliver is saying no.
No, we don't want raw data. We want your annotated spectra. Raw data is like, oh my gosh, who wants raw data? We have raw data. No, we want the your best opinion, and obviously these are opinions, right? You'd say, I think it's a galactosol, and you give a structure, and as you have shown in the very first part, sometimes we have to take some educated guesses by retention time or by other types of background knowledge, and then you give a structure. We have some in my opinion, as an author. That's what MassBank is, right? As an author, you have your name, and it's I think this spectrum is annotated with this structure. Right?
Now, by what we have in the supplementary materials, all the assigned fragments in the samples. So if you want to,
I'm really, nobody can read all papers, so things have to be electronically available, not in supplemental materials. So I would urge you and everybody else on the phone here on the call. It's like when you have a spectra, especially with spectra that are new spectra, right?
Yeah.
Just upload them to repositories and not to.
But also this information is not in the supplementary and also on GMPS. So everything is on GMPS. You can find also the supplemental Excel sheet, something is all in GMPS.
Okay.
So we really tried to make it accessible as good as we could. What we of course did not do is now, I mean, there's a list with all, no, we have that. That's basically a library. When you see this Excel, using this Excel sheet, you see all the fragments. This information is there, and we have examples in the supplementary how we did the assignment, so the fragment annotation. And also we tried, because that was a lot of, from the referees coming in, as you say, the educated guesses, we always tried to say whenever we were not sure what class it is. So we say based on that, we only saw one signal, and we are thinking this is that, but that's the limitation of the method.
Yeah, so, of course. You know.
I hope this.
I am. Yeah. Okay.
Okay, I'll go to the next question. Have you ever seen MGDG lipids in your studies, or in cell and human studies? We sometimes see them here at UC Davis in the feed lab, but they're only supposed to be in plants.
Yeah, good question. I saw them, of course, but in plants. So but I haven't searched from the MDGDs in the human samples. I would need to do that, because normally, because we are doing a lot with the decision rules, then we are saying we're searching for certain class, but when we're thinking it should not be where, we won't see it, and it's there, we won't see it.
Oh, I see. So it's more targeted.
Kind of like I mean, I could go now that you asked me and use the samples from the human lipid data analysis and just run it with the LDA for the different glycolipids. I can do that non-targeted. I just haven't done it because I don't expect them.
Yeah, exactly. So that's always like the untargeted way is we are open to everything, but also it means that we might be misled by something that looks like it. Like a betaine lipids look like a phospholipid, for example, right? And then you make a fool of yourself when you publish that and people laugh about you. So that's why we are hesitant to do that. On the other hand, just because somebody says it's not there, well, the idea of mass spectrometry is to say, well, the evidence is there, so we don't know where it comes from, but it looks like it's there, right? So to open your mind and not overlook elephants in the room just because some other people say, it can't be there, right? So that's the idea of untargeted analysis, really.
We could just run and let it run there and see if we could see something.
So if you
see it frequently, or I mean, maybe it's something
not frequently, but I think we saw them in brain.
Okay.
Yeah, so not frequently, but then I noticed that, and we're like, what is this doing there? Like, where does it come from? So I remember I talked to Shen, she's here on the call. It's like, is that even possible? And I think Shen looked at them and said they look like it, right? Yeah, so I'm just wondering, like, okay.
Especially because brain has this high ganglioside concentration, so the highest ganglioside concentration of all cells, that maybe there is some other class or this class doing something. I don't know.
Yeah, or it could be a breakdown. I don't know, right? Yeah.
Yeah, okay. Or it could be, yeah, as you said, there could be insert fragments or something. Let me think, is that possible with a ganglioside? No.
Not really, huh?
No.
It simply wouldn't be just because we eat plants, right? No,
you have to first of all, if we eat them, they have to be digested. You have to go through the endothelial layer. So that doesn't work in one way. So you have to disassemble and assemble, right? So that's not how it works.
Yeah, that seems complicated.
Usually, not for lipids, at least. At least not that I know.
Blood-brain barrier with
but
that's overrated anyway.
Yeah.
Okay.
Well, it's definitely something to investigate post a lecture here. So I'll go on to the next question. Why would amphiphilic lipids not show up at least a little bit in the methanol water part of the biphasic MTBE extraction?
Why won't the amphiphilic, so they're, okay, why they're separated?
Or
Good
they're not
question.
showing up?
Yeah, so what we basically did, we did the MTBE extraction and checked if all the gangliosides were there. And for us, we could not detect gangliosides in the methanol pathways based on our experience.
Yeah, but the amphiphilic part is like the idea is, well,
Yeah,
yeah,
I know,
right?
I know, but that's how it was.
Okay.
But yeah, maybe, I mean, people, there's also was always a referee question, and that's why we checked it. If there, I'm not, maybe the sialic acid part has something to do with it. I'm not sure. Yeah.
Yeah, you said they were acidic. How, like, what is their pH?
The pH of the gangliosides, depending on, yeah, depends now, because they can have 1, 2, or 3 sialic acids. What do you mean in solution, or?
But just in general, or like a range?
Yeah,
I thought that was pretty, pretty interesting information that I learned.
Yeah, no, I have to, sorry, that I have to look up again, but it has to be something. There was a paper on it and we looked into that, but I can't recall it now. I have to look it up, but it's a good question.
Okay. Sticking to the topic of extractions, are there any other sample preparation techniques that you've used other than MTBE to get more metabolite information or lipid information?
Yeah, as I said, for the plants, we had the mixture of isopropanol hexane water, and it really depends on the metrics we are using. But here's, of course, a lot of things we can still optimize. What we usually did in the beginning is we went to the literature, looked what the people did in order to analyze their different glycolipids, and then started with that. For example, and for the gangliosides, we wanted to do it with the MTBE because we wanted to do metabolites and lipids at the same time. And there it basically came out that we couldn't see it in the methanol part. But for the plants, you need something which is more harsher. So we have the isopropanol hexane. So we usually have different portions of either something which is isopropanol MTBE or hexane with water mixture at the moment. That's what we tested.
Okay, great. And
the alkaline hydrolysis was for the plants. It's not working otherwise because this starch thing is just crazy sticky. It's just everywhere. Okay. Yeah. So if you do the plants to get rid of that.
One last question here. How are glycolipids biologically important in humans versus non-glycosylated lipids?
How they're important versus non-glycosylated lipids. Okay, yeah, because so the examples, I mean, there are different glycolipids out there. I focus here on the glycosphingolipids, which are the ones which are inside the membrane bound with the lipid part and outside the membrane pointing outside from the cell. So in terms of biology, compared to a standard lipid, for example, the phosphatidylcholine, which is just inside the membrane, it has this glycan part outside, which is responsible for signaling. So also, a lot of studies have shown when we do bulk analysis that there are not a lot of differences, but for the glycolipids, they're super interesting because they're involved in all the signaling pathways. So in terms of biology, I would say the examples for glycolipids of the glycosphingolipids are mainly with the signaling difference in the difference compared to the standard bulk lipids. So I don't know how pathogen interaction, for example, it was shown that for COVID, it's the gangliosides are entering point or helping for the signal entering the virus. There are all kinds of things just discovered now with the different glycolipids. So they're super important. Post-pathogen interaction signaling compared to things which are more inside the cell membrane or inside the cell. Yeah.
Great. Those are the last questions in the chat. can open it up. We still have some time for questions if participants have them. You can unmute yourself or keep adding to the chat. I guess not. Well, thank you, Dr. Rampler, for such a great presentation. And also, giving this talk while you're on maternity leave, we really appreciate it. Yeah, that's really wonderful.
Thanks for having me. It was very interesting. And if you have any follow-up questions or you want to try this and need something with the data, just let us know. It would be great.
Yeah, of
and
course.
I'm looking at the MTDT part in human.
Yeah, wonderful. Okay, I'd like to share a little bit about our next speaker before we all head out. So our next speaker is Professor Kelly Marie Hines from University of Georgia. She'll be talking about ion mobility mass spectrometry for high throughput omics analysis. Specifically, her lab, I think, also focuses on antibiotic resistance studies. So yeah, if you're interested in that, please join us on December 7th. Same time, same place. So again, yes, thank you for joining, and we'll see you next month.